Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Dr. Samir Kalgatki, Director of Molecular Biology for WuXi Advanced Therapies.
Thank you so much, Alex. I'm really looking forward to this exciting webinar where I will discuss robust methods for stability and identity testing for novel cell and gene therapies. So in this presentation, we will discuss the need for stability and identity testing and the challenges associated with this kind of testing. For novel cell and gene therapy products to gain regulatory approval, it has to go through a genetic stability and identity testing, and these tests should pass for the FDA approval to be obtained. The three major technologies that are used for determining stability and identity of the various cell and gene therapy products are Sanger sequencing, human cell line identification using STR or short tandem repeats, and next generation sequencing. In this webinar, you will learn about the considerations when planning the genetic stability and identity testing for novel cell and gene therapy products. I will walk you through in a high-level overview of how these methods work and the type of samples on which they can be performed. And finally, I will describe how WuXi applies proprietary protocols to support customers in their stability and identity testing needs. I will start first with next generation sequencing, or NGS for short. WuXi offers whole genome sequencing of viral genomes, which could be AAV vectors or lentiviral vectors. We typically sequence viral suspensions. In addition, we can also sequence starting material that are listed in the table below, for example, HSV, baculovirus, CMV, and or plasmids, which can serve as an identity assay. The process starts by preparing a fragment library of the client product, which could be a lentiviral vector or a AAV vector. The fragment library is prepared following by sequencing of the entire product. WuXi uses the Ion Torrent Genexus platform from Thermo Fisher for our next generation sequencing assays that include whole genome sequencing, targeted sequencing, and cell line identification for non-human origin products. The platform is an automated library preparation, templating, and sequencing all in one instrument. Because it is totally automated, it reduces the turnaround time and decreases operator errors. The instrument allows to run 32 samples at once in a batch run. The total reads that are generated per run are 64 million, and the read length is 200 to 300 base pairs. So you can increase the reads per sample by decreasing the number of samples that are run in a single batch. So if we run, for example, 2 samples only, then each sample will have 32 million reads. So depending on the stringency and depending on the needs of the client, we can increase or decrease the number of reads per sample. This is a flexibility that several clients would really enjoy and would love to have. Very quickly, I'll walk through the workflow for the NGS method for whole genome sequencing of vectors or plasmids. It starts by extraction of genomic DNA or RNA. In the case of RNA, we ask that the RNA go through a cDNA preparation. Once we have genomic DNA or cDNA, Both of these kind of DNAs undergo fragmentation, followed by ligation of adapters and barcodes. These samples are then input into the ion-torrent Genexus platform that performs the templating and sequencing. Once the sequencing run is completed, it is followed by bioinformatics data analysis. In this case, WuXi uses its own proprietary bioinformatics pipeline to conduct the data analysis. The sequence obtained from the sequencing run is aligned to the client-provided reference sequence, and any variance between the sequence sample and the client-provided reference sample are reported to the client. To elaborate a little bit of the bioinformatics platform, I'm providing an example of a specific virus sample that was sequenced at WuXi. The pipeline aligns the sequence data to the client reference, as I mentioned in my earlier slide. The consensus sequence, the coverage map, and any identified variants are reported. In this particular example, if you pay attention to the table below the plot, we can see after sequencing and bioinformatic analysis was completed, the pipeline identified 3 SNPs or single nucleotide polymorphisms, which basically means the reference sequence had a nucleotide T at position 817, but after sequencing was completed, we found out that there was actually the presence of nucleotide C. There was only one, and that's why it's a single nucleotide polymorphism. In total, the pipeline identified 3 SNPs and 2 indels. Indels are insertions or deletions. In this case, at position 1632 and position 5198, there was a deletion of nucleotide C. The default criteria for a base to be called is QV greater than or equal to 20. What that means is a 99% accuracy. For every 100 base pairs read, there may be 1 error, and the read depth has to be greater than 15. All of this information is provided to the client in a report after completion of the bioinformatic analysis. This is how we use NGS for whole genome sequencing that could be applied to lentiviral vectors or AAV vectors, and it can also be applied to the helper material that is used in manufacturing of novel cell and gene therapy products. The next use of how we use NGS or next generation sequencing for helping our cell and gene therapy clients is to help them perform cell line identity. In this case, we offer non-human cell line identity using next generation sequencing on the ion torrent. The process consists of targeted amplification and sequencing of 10 to 15 species-specific target or loci to confirm the cell line identity. These loci are highly conserved and well-defined for different species. So we can use these loci, and the presence and absence of at least 12 out of the 15 loci can tell us about a match or a mismatch of the identity. Typical sample types that we accept for this sample and for this kind of assay is either a frozen cell pellet or a frozen cell suspension. We require a minimum of 10 to the 6 cells. There are different cell lines that we have reference sequences for that can be used for identity testing. Some of them include the Chinese Hampshire ovary, SF9, Vero cells, and or BHK cells. The workflow for the identity assay works in many cases similar to the whole genome sequencing. It starts by extraction of genomic DNA. For cell line ID, we only sequence genomic DNA. It's followed by targeted amplification. So multiple sets of forward and reverse primers amplify the 10 to 15 different regions of the genome. Then these sequences are ligated with adapters and barcodes. The sample is then inputted to the ion torrent for templating and sequencing. After templating and sequencing is completed, it undergoes the bioinformatic analysis using WuXi's proprietary pipeline. The reads are aligned to species-specific target sequences. And as I mentioned, as long as there are 12 out of the 15 loci are reference matched, we are able to confirm the identity of the cell line. Here is an example of what the report looks like for a Chinese hamster identity assay. There are 15 loci that are well defined by our WuXi's pipeline and method development. We show the target, the coverage that we obtained. We performed blast of each of these loci against the Chinese hamster ovary genome, and we provide the result. In this particular example, as you can see, we have a 100% match for each of the 15 loci that are probed in the identity assay. All 15 of 15 Chinese hamster ovary loci were detected, and the result is identity confirmed. The WuXi Advanced Therapies pipeline aligns the sequence data in a species-specific manner. The sequences are called positive if they have a greater than equal to 80% of the loci spats, which is what I was telling earlier. 12 out of the 15 loci need to match. And the blast hit, the top blast hit, should be for the species being tested. So in this particular example, we found out that the top blast hit was the Chinese hamster ovary as expected. So the whole genome sequencing of viral vectors or helper material and cell line identity of non-human cell lines are two assays that our clients can use for obtaining a confirmed match of the identity and also determining the genetic stability of the viral vectors. I'm then going to move on to next technology that can also be used for sequencing of the viral genomes and or for confirming identity. So Sanger sequencing is another method that WuXi offers to its clients. Sanger sequencing is widely used for testing genetic stability and determining the identity of specific target regions in cell and gene therapy products. Sanger sequencing is a gold standard method. Other common applications of Sanger sequencing also includes the confirmation and filling the gaps in the results of next generation sequencing. The sample types that we accept are varied. They can be cell banks, they can be frozen cell pellets, bacteria in cell pellets or aliquots, purified plasmid DNA, and or virus seed stocks and samples. So WuXi ATU proprietary protocols are applied to sequence difficult regions like high GC rich regions, hairpin secondary structures, and also for confirmation of homopolymer repeat regions, which can sometimes be difficult for next generation sequencing based methods to resolve. The Sanger sequencing method has three simple steps. It has several advantages. It's a gold standard method for sequencing. The reaction can be set up in a single tube. It has a high degree of accuracy. It has the ability to sequence long fragments. If you saw my previous slides, NGS offers 200 to 300 base pair sequencing, whereas Sanger sequencing can do thousands of base pairs. It offers a high confidence of single nucleotide polymorphism detection, and it can also resolve homopolymer regions. The samples are loaded into the ABI genetic analyzer. The genetic analyzer obtains a chromatogram as shown here in the image. A software then converts the chromatogram into reads. The reads are then aligned in reference to the sequence that was provided by the client. It does it in a nucleotide by nucleotide approach. If any single nucleotide polymorphisms or any insertions or deletions are determined, the software alerts the user for the presence or absence of the nucleotides, and or the replacement of the reference nucleotide with another nucleotide after sequencing is completed. The reference-based assembly allows us to offer a robust, high-quality method for clients to determine their sequence or to determine the stability of the sequence and or to determine the identity of their sample. To go into a little bit more detail about how the Sanger sequencing process works, I'm going to walk through quickly the workflow. The PCR amplified target regions in the genomic DNA or the complementary DNA generated from total RNA is used for the Sanger sequencing. Sanger sequencing can also be performed directly on the purified plasmid. It starts by extraction of genomic DNA or extraction of total RNA. In the case of total RNA, it undergoes a cDNA synthesis, and then in this case, the extracted genomic DNA or the cDNA undergoes PCR amplification of targeted regions using multiple pairs of forward and reverse primers. The sequencing reaction is performed in the single tube. The sequencing performs on the ABI using the samples that were obtained, and then the chromatogram is generated by the sequencer, and the reads are aligned in a reference match manner. The data analysis is performed on our proprietary pipeline, and the results are shared with the client as the obtained sequence is identical to the reference or if any discrepancies were found. A similar approach is for plasmid purification. Pure plasmids can also undergo Sanger sequencing. Specific regions of the plasmid are amplified using PCR primers that are targeted to that region. The sequencing reaction is completed, followed by sequencing on the ABI. The ABI performs the sequencing and provides a chromatogram, and then the assembly of the reads obtained is performed using WuXi's data analysis pipeline, followed by a report. STR analysis using Sanger can also be used for human cell line identification. I will walk you through the cell line identification assay using STR analysis. Short tandem repeat analysis, or STR analysis, is a method that is a PCR-based method to identify human cells. All of our human genomes have specific STRs that make us all unique. These STRs can be anywhere from 7 to 12 repeats. To identify a specific cell line, we amplify 10 to 15 different STR sites. These STR sites are well defined. The loci are distributed throughout the human genome and are highly polymorphic. The loci are amplified by PCR and analyzed with size standards. Here is an example of what those loci look like and how many repeats did those loci have. WuXi Advanced Therapies offers STL analysis for determining identity, but the procedure is limited to human cell lines only. The PCR amplification and fragment analysis is used to generate the STR DNA profile for up to 10 loci. The ATCC Standards Development Organization work group recommended that such identity assay have at least 8 or more STR loci. The STR DNA profile is generated for the cell line on the ABS 3500 XL genetic analyzer. It is then used to authenticate human cell lines by comparing to reference sequences on the ATCC database. The comparison between the reference sequence and the profile obtained from the sample can be performed using this assay. Alternatively, we can have a client-provided reference sequence, and then the reference sample is used as the reference sequence to match the product sequence. So we could either match it to the ATCC database or to a client-provided reference sequence. Just giving a quick overview of the STR data analysis, method. Samples used are primary human cells. The minimum cell count is 10 to the 6 cells. The cell pellet or the cell suspension is isolated to obtain genomic DNA. Multiple PCR reactions are performed that amplify the different loci. We have several sets of reverse and forward primers. The PCR fragments so obtained then undergo capillary electrophoresis and fragment analysis on the ABI 3500 exogenetic analyzer. We have the STR profile generation in the middle of the slide here. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different loci that are amplified. You can see very clearly that some loci are homozygous, which means they were obtained from just one parent. Any loci that has two peaks are heterozygous. That means they were obtained from both parents. And in this case, we then use these peaks to compare to the reference profile between the client sample and the reference from the ADCC's database. If there is a match, we determine how much match. We have set 80% as the threshold, as the minimum number of loci that need to match for us to call the comparison as identical samples. When we compare to the ATCC database, we get several hits, and we want to ensure that the top hits are human. In this case, we can see that there were 2 cell lines that were 100% match, and then there was a third cell line which was an 89% match. Because the threshold is 80%, we report all 3 cell lines from the ATCC database as a potential match to the client's reference sequence or to the client sequence obtained at WuXi. In summary, the stability and identity testing for cell and gene therapy are conducted using Sanger or NGS sequencing. WuXi offers these 3 methods for clients that can help confirm the stability and identity of their viral vector products. For regulatory approval, the identity and stability of unspected DNA inserts must be confirmed. Sanger sequencing is widely used for this purpose and also to confirm the results of next generation sequencing, especially for hard to sequence regions like homopolymer repeats. STR analysis can also be used to authenticate if cell lines are of human origin and to verify the identity of cell lines used in viral vector manufacturing. Finally, high-throughput sequencing can be used for whole genome sequencing of lentiviral and AAV vectors, or to identify cell lines used to manufacture such novel cell and gene therapy products. I thank you so much for your attention, and I would be happy to take any questions. Thank you once again.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, which kind of samples can be processed by WuXi on these methods?
So as I mentioned earlier, the methods are robust and are flexible and can sequence different samples. They can definitely sequence for whole genome sequencing lentiviral and adeno-associated viral vectors. We can also sequence the starting material used for the manufacturing of such viral vectors. For example, herpes simplex virus, baculovirus, or CMV. As far as the sample types are concerned, we can sequence cell banks, we can sequence viral suspensions, we can sequence the helper material used to manufacture these viral material, and we can also sequence master cell banks, working cell banks, end of process cycle cell banks. We have the ability to help the clients sequence their whole pipeline for manufacturing of cell and gene therapy products.
And how should clients decide between NGS and Sanger?
That's a great question. Sanger sequencing is definitely the gold standard. But next generation sequencing allows you to obtain whole genome information for larger sequences. Typical viral vectors are anywhere between 5 kb to 10 kb. Uh, Once we reach the 3000 base pairs, the cost and time benefit between Sanger and NGS start making NGS more economical and more viable. But Sanger sequencing is still used when we want to obtain sequence where the sequence length is between 1000 and 3000 base pairs. Sanger sequencing definitely makes more sense at the smaller or shorter genome length. And finally, Sanger sequencing is also helpful when next generation sequencing is unable to resolve certain hard to sequence regions like homopolymer repeats. At the end of the day, the clients really need to make a call of which method makes the more sense. WuXi will definitely be able to provide technical support in helping them come with this decision, but the client should be the one making the most uh, conscious decision using all of the information that WuXi can provide. And this also needs to include a discussion with the regulatory person that the client is working with to ensure that the regulatory approval is obtained by providing data from the best method that is applicable to their sample.
So it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Samir, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, why is the 80% match criteria chosen by WuXi? Why does the STR assay have 13 loci for identity analysis?
So let me start by answering the first question, which is why is 80% chosen as a threshold for determining if the sample is a match to the reference sequence. So the reference sequence in the PubMed database are not identical, and a certain amount of flexibility is required to handle the variations in either the CHO cell lines or non-human cell lines and or human cell lines. The 80% criteria is based on International Cell Line Authentication Committee's guide to human cell line authentication that was published on 9 January 2014 for short tandem repeat analysis. It, it recommends that we should apply an 80% match threshold to allow for variation in cultured samples. Using this guideline, WuXi also stayed with the 80% as the threshold for reporting a match or not a match between the reference sequence and the client provided sample. For the second question, which is why we have chosen 15 loci, the assay was developed in 2015, and we wanted to make sure that the method stays consistent with other methods available out there that also use 15 targets for cell line identity assays. The species identification in cell culture is a two-pronged approach. The method expanded to target more genes, exceeding the number of targets analyzed for in the isoenzyme or the STR test. And this allows us to provide clients a very robust method where they can take the results and use these results for regulatory submissions. Because it is the 80% threshold, we need to show at least 12 out of the 15 loci match. This allows us to provide confidence to clients that their sequence obtained after manufacturing campaign is a match to the reference sequence, and they can continue on with further manufacturing without worrying about the genetic stability of the product and or the identity of the cell line that was used to manufacture their product. This is very critical for clients because at the end of the day, these cell and gene therapy products are going to be administered to patients and allow them to have a better way of life without having to worry about the safety of the drug that was infused or injected in them.
Okay, great. Well, thank you, Samir.
You're most welcome,
And
Alex.
thank you
Thank you
to
so much.
our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.